Hello, good afternoon, everybody. It's my sincere hope that you're doing fine. I'm well. This afternoon, I want us to take some time to go through the parts of a light microscope. I want us to have an overview, and I want us to study the parts or the structural parts of the microscopes together with their functions. So, as you can see from the notes on your screen, the microscope was first manufactured in the 16th century. Microscopes have revolutionized science with the ability to magnify small objects such as microbial cells, producing images with definitive structure that are identifiable and characterizable. So, what are microscopes, if we may ask? Microscopes are instruments that are used in science laboratories to visualize very minute objects such as cells, microorganisms, giving a contrasting image that is magnified. Microscopes are made up of lenses for magnification, each with their own magnification powers. Depending on the type of lens, it will magnify the specimen according to its focal strength. Their ability to function is because they have been constructed with special components that enable them to achieve high magnification levels. They can view very small specimens and distinguish their structural differences. For example, the view of animal and plant cells. Viewing of microscopic bacterial cells also is something that you do using a microscope. It could be electronic or it could just be the normal light microscope. So microscopes are generally made up of structural parts for holding and supporting the microscope and its components, and the optical parts which are used for magnification and viewing of the specimen image. So what you're saying here is that you can classify the parts or the structural components of a microscope into two: the structural parts that enable the holding and supporting of the microscope itself and the components, and the second part is the optical part, which is used for magnification and viewing of the specimen images. This description defines the parts of a microscope and the functions they perform to enable visualization of the specimen. And I want us to take a look at the diagram that is on your screen here, and you will realize that, as we just said, there is the structural part of the microscope, which is basically the arm, or maybe the structure itself. And then you've got this bit down here which plays a big role in the magnification of a purpose of a microscope. So as you can see from the listing, there is the ocular lens, or what is called the eyepiece. There is the diopter adjustment for adjusting the eyepiece, the nosepiece, objective lens, stage clip, you got the aperture, you got the diaphragm and condenser. Of course, these are parts of the optical part for magnification. You got the illuminator that supplies the light, and then you have the head, the arm, or if you may, the carrying handle, the mechanical stage, the coarse adjustment for adjusting the specimen that you're viewing. Then you got the fine adjustment, the stage controls, the base, the brightness adjustment, and light switch, the light switch. So basically those are the parts of a microscope. So if you look at the functions of each one of those parts, starting with the head, which is also known as the body, it carries the optical parts in the upper part of the microscope. The base acts as the microscope support and carries the microscopic illuminators. By illuminators, we mean those components that are supplying light. Then you have the arms. This is the part, or the arm rather, is the part connecting the base to the head and the eyepiece tube to the base of the microscope. It gives support to the head of the microscope, and it's also used when carrying the microscope around. Some high quality microscopes have an articulated arm with more than one joint, allowing more movement of the microscope's head for better viewing. By articulated, we mean it can move in many different ways. It's kind of jointed. So if you look at the optical parts of a microscope together with their functions, you'll notice that the optical parts of the microscopes are used to view, magnify, and produce an image from a specimen placed on the slide. These parts, the optical parts, will include the eyepiece, also known as the ocular. This is the part used to look through the microscope and is found at the top of the microscope. Its standard magnification is normally 10 times, with an optional eyepiece having magnifications of 5 times to 30 times. By that we mean if you place a specimen on a slide and observe it, the standard magnification of what you're going to see is 10 times the original size. Then you got the eyepiece tube. It's the eyepiece holder. It carries the eyepiece just above the objective lens. In some microscopes, such as the binoculars, the eyepiece tube is flexible and can be rotated for maximum visualization. You can adjust it to suit your sight. For variance in distance, for monocular microscopes, they are non-flexible. That's to say, they're fixed. You cannot adjust them. And you got the objective lens. These are the major lenses used for specimen visualization. They have a magnification power of 40 times to 100 times. Oh, that's powerful. They can multiply what you're going to see from its original size to even up to 100 times, meaning you can see a lot of detail. There are about 1 to 4 objective lenses placed on one microscope, in that some are rear facing and others face forward. By rear facing, we mean they face backwards, and others will face the front. If you look at the nosepiece, also known as the revolving turret, it holds the objective lenses. It is movable, hence it can revolve the objective lenses depending on the magnification of the lens. The nosepiece, we are simply saying this part here, the nosepiece can be rotated. It can go that direction. It can also go this direction to suit the observation that you want to make. Let's now go ahead and look at the adjustment knobs. These are knobs that are used to focus the microscope. There are 2 types of adjustment knobs. There are fine adjusters and the coarse adjustment knobs, meaning with one set you can do some coarse, really rough adjustment when it's quick, and then you can fine-tune with the second set. The stage is the section on which the specimen is placed for viewing. They have stage clips to hold the specimen, of course, and hold the slide in place. The most common stage is a mechanical stage, which allows the control of the slides by moving the slides using the mechanical knobs on the stage instead of moving it manually with your hands. So you also got another part called the aperture. It is a hole on the microscope stage through which the transmitted light from the bottom from the source now reaches the stage, and it's able to illuminate your specimen. Microscopic illuminator. This is the microscope's light source. It's located at the base. It is used instead of a mirror. It captures light from an external source of a low voltage of about 100 volts. And this is actually what we're talking about, the illuminator. This is the source of light from the bottom of the microscope, and it sends light right through there and illuminates the specimen, which is here. Now let's go ahead and look at the condenser. These are lenses that are used to collect and focus light from the illuminator into the specimen. They are found under the stage next to the diaphragm of the microscope. The diaphragm is somewhere beneath the stage. It is somewhere beneath the stage. The diaphragm should be here. This is the stage, is what we are calling the stage, and this is the diaphragm. It's right below the stage. And we are saying the diaphragm, which is also known as the iris. It's found under the stage of the microscope, and its primary role is to control the amount of light that reaches the specimen. It is more or less like the iris in your eye. It can open and it can kind of close to allow more light or less light depending on what you need. It is adjustable, hence controlling the light intensity and the size of the beam of light that gets to the specimen. For high quality microscopes, the diaphragm comes attached with an Abe condenser, and combined they are able to control the light focus and light intensity that reaches the specimen. Now the next part of the microscope that you want to look at is the condenser focus knob. This is a knob that moves the condenser up or down, thus controlling the focus of light on the specimen. Then the Abe condenser is a condenser specially designed on high quality microscopes, not on every microscope, and it makes the condenser to be movable and allows very high magnification of above 400 times. The high quality microscope normally have a high numerical aperture than that of the objective lenses. Then the rack stop, the rack stop is a part that controls how far the stage should go, preventing the objective lens from getting too close to the specimen slide, which may damage the specimen. It is responsible for preventing the specimen slide from coming too far up and hit objective lenses. So we are saying that they are placed somewhere here, the stage clips, and then we're looking at the rack stop that is meant to make sure that the slide, which of course you'll place somewhere here in the middle, because you'll always place your slide somewhere here, and you don't want them coming too close to the objective lens, and you don't want the objective lens damaging whatever will be on your specimen. So you have the rack stops to control the movement of the slide. And basically that is what we might have as an overview of a microscope. Now there are some review questions that you want to think about, maybe to test your knowledge. You may want to define a microscope, to state its functions, maybe diagrammatically identify the various parts of a microscope, then describe the functions of each part of the microscopes you have drawn, and differentiate between a condenser and an Abe condenser. What is the magnification power of the objective lenses? How does the eyepiece compare to the objective lens? You may also want to ask yourself, why is the rack stop included in the microscope from the factory, and whether it can be replaced. Then what is a magnification power? Then from there probably you may want to differentiate between the fine and coarse adjustments knobs. So as of today, we're going to end our lesson there, and I hope it was good for you. Now you understand the structure and the functions of the various parts of a microscope.